Neuromuscular transmission. Skeletal muscle fibers are innervated by large myelinated nerve fiber that originates from large motoneurons in the anterior horns of the spinal cord. Each nerve fiber, after entering the muscle belly, normally branches and stimulates 3 to several hundred skeletal muscle fibers. Each nerve ending makes a junction called neuromuscular junction with the muscle fiber near its midpoint. The action potential initiated in the muscular fiber by the nerve impulse travels in both directions towards the muscle fiber ends. With the exception of about 2% of the muscle fibers, there is only one such junction per muscle fiber. This is the introduction of the neuromuscular transmission. Now we comes to the physiological anatomy of the neuromuscular junction. Physiological anatomy of neuromuscular junction, the motor end plate. Neuromuscular transmission. This topic is presented before you by me, myself, Prem Singh Chauhan, Associate Professor in Zoology at Set Ranglal Kothari Government PG College, Rajasthan. First of all, we look at this diagram. This diagram explains this is axon. Axon is covered by the Schwann cell, so it is the myelin sheath over the axon. This terminal portion of the terminal nerve branch, teloglial cells, and this one is the portion of the muscle fiber. It consists of muscle nucleus, and this long one is myofibril. This diagram again explain the surface view of the end plate. This one is the surface view of the end plate. And the third one diagram, this third one diagram explain the electron microscopic appearance of the contact point between the single axon terminal and muscle fiber membrane. This terminal of the axon enlarge in button shape, which form axon terminal in synaptic through. There are so many synaptic vesicles filled in it, and these are the mitochondria present at the motor end plate. But it says that this diagram shows the neuromuscular junction from a large myelinated nerve fiber to a skeletal muscle fiber. The nerve fiber forms a complex of branching nerve terminals that innervates into the surface of the muscle fiber but lies outside the muscle fiber plasma membrane. The entire structure is called the motor end plate, and it is covered by one or more Schwann cells that insulate it from the surrounding fiber. The invaginated membrane is called the synaptic gutter or the synaptic throat. This one is synaptic throat. And the space between the terminal and the fiber membrane is called the synaptic space or the synaptic cleft. This is the space between these two axon terminal synaptic through and muscle fiber membrane. So this space is known as synaptic cleft, CLEFT, synaptic cleft or the synaptic space. This space is 20 to 30 nm wide. At the bottom of the gutter, the numerous small folds of the muscle membrane called subneural clefts. These are sub subneural cleft, which greatly increases the surface area at which the synaptic transmitters can act. In the axon terminal are many mitochondria. These are the mitochondria. At the axon terminal are many mitochondria that supplies ATP, adenosine triphosphate, the energy source that is used for the synthesis of an excitatory transmitter, acetylcholine. The acetylcholine in turn excites the muscle fiber membrane. Acetylcholine is synthesized in the cytoplasm of the terminal, but it is absorbed rapidly into the small synaptic vesicles, about 300,000 of which are normally in the terminals of a single end plate. The synaptic space are large quantities of the enzyme acetylcholine esterase, which destroy acetylcholine a few milliseconds after it has been released from the synaptic vesicle. These small vesicles are synaptic vesicles. These synaptic vesicles consist of acetylcholine, this acetylcholine is synthesized in the synaptic cleft where there is a enzyme present which is known as acetylcholine esterase. This acetylcholine esterase acts upon the acetylcholine and destroy the acetylcholine within the few milliseconds. Secretion of acetylcholine by the nerve terminals. When a nerve impulse reaches at the neuromuscular junction, about 125 vesicles of acetylcholine are released from terminals into the synaptic space. Some of the detail of this mechanism can be seen in this figure presented before you in this slide. So these are the release sites of the acetylcholine. This one is neural membrane and these are the vesicles of the acetylcholine. This is dense bar, and red one showing the calcium channels. Basal, lamella and acetylcholine are present in between them. There are few acetylcholine receptors are present on the membrane of the muscle fiber. So these are the acetylcholine receptors with voltage activated sodium channels. These are the voltage activated sodium channels, and this one is muscle membrane. So it shows an expanded view of synaptic space with the neural membrane above and the muscle membrane and its subneural clefts below. On the inside of the surface of neural membrane are linear dense bars. Shows in this view to each side of each dense bar are the protein particle that penetrates the neural membrane. These are voltage gated calcium channels, and these voltage gated calcium channels are present here and demonstrated by two red bars. These are the calcium channels. Voltage gated calcium channels. When an action potential is spread over the terminal, these channels opens and allow calcium ions to diffuse from the synaptic space to the interior of the nerve terminal. As and when action potential comes to this membrane, this calcium channel opens and this calcium channel absorb the calcium from the synaptic cleft to the nerve terminal. In this direction, these are the calcium channel which receives calcium from the synaptic cleft. The calcium ions are believed to activate the calcium calomodulin dependent protein kinase, which in turn phosphorylates synapsin protein that anchor on the acetylcholine vesicle of the cytoskeleton of the presynaptic terminal. This calcium ions activate the calomodulin dependent protein kinase. This calomodulin dependent protein kinase is present on the surface of the vesicles of the acetylcholine. This in turn phosphorylates synapsin protein. Synapsin protein that anchors on the acetylcholine vesicle to the cytoskeleton of the presynaptic terminal. And this process frees the acetylcholine vesicle from the cytoskeleton and allows them to move to the active zone of the presynaptic neural membrane adjacent to the dense bars. The vesicles then dock at the release site, fuse with the neural membrane, fuse with this is the fusion phosphorylation of the cytoskeleton releases this vesicle, and this vesicle moves towards the surface of the neural membrane and then release their content acetylcholine into the synaptic cleft. These vesicles then dock and release sites, fuse with the neural membrane and empty their acetylcholine into the synaptic space, by the process of exocytosis. This has been completed by the process of exocytosis. Although some of the aforementioned detailed are specular, it is known that effective stimulus for the causing acetylcholine release from the vesicle is entry of the calcium ion, and the acetylcholine from the vesicle is then emptied through the neural membrane ascend to the dense bar. Now, the acetylcholine opens ion channel on the postsynaptic membrane. This acetylcholine enters into the synaptic clefts and it opens ion channels of the postsynaptic membrane, which is the membrane of the muscle fiber. Many small acetylcholine receptors in the muscle fiber, these are the acetylcholine receptors present on the surface of the muscle fiber membrane, and these are acetylcholine gated ion channels and are located almost entirely near the mouth of the subneural cleft, laying immediately below the dense bar areas where the acetylcholine is empty into the synaptic space. The channel remains constricted acetylcholine opens. This is iron, not iron. Ion channels on the postsynaptic membranes. This figure shows the channel. When channel remain constricted until two acetylcholine molecule attach to the receptor. This attachment causes a conformational change that opens the channel. The two acetylcholine molecule, this is acetylcholine molecule, attach at the surface of the channel, and then this channel protein undergoes conformational change, so it opens channel for the sodium ion that enters into it. The acetylcholine gated channel has a diameter of about 0.65 nanometer, which is a large enough to allow the important positive sodium ion, potassium and calcium to move easily through the opening. The patch clamp studies have shown that one of these channels, when opened by acetylcholine, can transmit 15,000 to 30,000 sodium ion in a 1 millisecond. The capacity of this channel is to allow 15,000 to 30,000 sodium ions within 1 millisecond. Conversely, negative ions such as the chloride ions do not pass through because the strong negative charges in the mouth of the channel that repels these negative ions. This is the negative charges which repels the chloride ions and do not allow the chloride ion to diffuse through it. In the practice, far more sodium ions flow through the acetylcholine gated channels than any other ions for two reasons. First, there are only two positive ions in the large construction, sodium ions in the extracellular fluids, and potassium ions into the intracellular fluid. Second, the negative potential on the inside of the muscle membrane, minus 80 to minus 90 millivolt, pulls the positively charged sodium ion to the inside of the fiber, while simultaneously preventing efflux of the positively charged potassium ions when they attempt to pass outward. I repeat it once again that there is a for far more sodium ions flow through the acetylcholine gated channels than any other ions for two reasons. First, there are only two positive ions in the large concentration. Sodium ions are present in the extracellular fluid and potassium ions are present into the intracellular fluid. Second, the negative potential inside the membrane minus 80 to minus 90 millivolt. This negative potential pulls the sodium ions towards the intracellular fluid, while simultaneously it prevents efflux of the positively charged ion potassium ions when they attempt to pass outward. This 12.3, this B1 diagram shows the principal effect of the opening of the acetylcholine gated channels is to allow large number of sodium ions to pour to inside of the fiber, carrying with them large number of positive charges. This action creates a local positive potential inside the muscle fiber. The entry of the large number of sodium ion creates positive charges in the inner side of the membrane, and this action creates a local positive potential that change inside the muscle fiber membrane called the end plate potential. In term end plate potential. In turn, this end plate potential initiates an action potential that is spread along the muscle membrane and thus cause muscle contraction. Destruction of the released acetylcholine by acetylcholinesterase. So we reaches here 1, 2 completed end plate potential and excitation of the skeletal muscle fiber. Before we study this one now, destruction of released acetylcholine by acetylcholinesterase. Acetylcholine, once released into the synaptic space, continues to activate the acetylcholine receptors as long as the acetylcholine persists in the space. However, it is removed rapidly by two means. First, most of the acetylcholine is destroyed by the enzyme acetylcholinesterase, and a small amount of acetylcholine diffuses out of the synaptic space and is then no longer available to act on the muscle fiber membrane. The short time that the acetylcholine remains in the synaptic cleft, a few milliseconds at most, normally is sufficient to execute the muscle fiber. Then the rapid removal of the acetylcholine prevent continuous muscles re-excitation after the muscle fiber has recovered from its initial action potential. The end plate potential and excitation of the skeletal muscle fiber, the sudden insurgence of the sodium ions into the muscle fiber, when the acetylcholine gated channels open, cause the electrical potential inside the fiber at the local area of the end plate to increase the positive direction as much as 50 to 75 millivolt, creating a local potential called the end plate potential. A sudden increase in the nerve membrane potential of more than 20 to 30 millivolt is normally sufficient to initiate more and more sodium channels opening, thus initiating an action potential at the surface of the fiber membranes, muscle fiber membrane. Here is one diagram. This diagram. This shows the principle of end plate potential initiating the action potential. This figure shows three separate end plate potentials. End plate potentials A and C. These three end plate potential A, B and C. This A and C are too weak to elect an action potential, but they do not produce weak local end plate voltage changes as recorded in the figure. By contrast, end plate potential of the B is much stronger and cause enough sodium channels to open so that the self-regenerative effect of more and more sodium ions flowing to the interior of the fiber initiates an action potential. Safety factor safety factor for transmission at the neuromuscular junction, fatigue of the junction. Ordinarily, each impulse that arrives at the neuromuscular junction causes about 3 times as much end plate potential as that requires to stimulate the muscle fiber. Therefore, normal neuromuscular junction is said to have a high safety factor. However, stimulation of the nerve fiber at rates greater than 100 times per second for several minutes often diminishes the number of acetylcholine vesicles so much that impulse fail to pass into the muscle fiber. This situation is called fatigue of the neuromuscular junction, and it is the same effect that causes fatigue of the synapse in the central nervous system when the synapses are overexcited. Under normal functioning condition, measurable fatigue of the neuromuscular junction occurs rarely, and even then only at the most exhausting levels of muscle activity. Molecular biology of the acetylcholine formation and release. Formation and release of the acetylcholine at the neuromuscular junction occurs in following stages. The first stage, there is a small vesicles about 40 nm in size are formed by the Golgi apparatus in the cell body of the motor neuron in the spinal cord. These vesicles are then transported by the exoplasm all the way to the neuromuscular junction at the tip of the peripheral nerve fiber. About 300,000 of these small vesicles collect in the nerve terminals of a single skeletal muscle end plate. So the size of the small vesicles is 40 nm and the number is 300,000 small vesicles collects at the nerve terminals. Second, the acetylcholine is synthesized in the cytosol of the nerve fiber terminal but is immediately transported through the membrane of the vesicle to their interior where it is stored in highly concentrated form, about 10,000 molecules of acetylcholine in each vesicle. Each vesicle consists of 10,000 molecules of acetylcholine. When an action potential arrives at the nerve terminal, it opens many calcium channels in the membrane of the nerve terminal because the terminal has an abundance of voltage gated calcium channel. As a result, the calcium ion concentration inside the terminal membrane increases about 100-fold, which in turn increase the rate of fusion of the acetylcholine with the terminal membrane about 10,000 fold. This fusion makes many of the vesicles rupture, allowing exocytosis of the acetylcholine into the synaptic space. About 125 vesicles usually rupture with each action potential. Then after a few milliseconds, the acetylcholine is split by the acetylcholine esterase into the acetate ion and choline, and the choline is reabsorbed actively into the neural terminal to be reused to form new acetylcholine, and this sequence of the events occur within a period of 5 to 10 milliseconds. Few important points I would like to repeat. When the action potential arrives at the nerve terminal, it opens many calcium channels. So the absorption of the calcium ions from the synaptic cleft to nerve terminal takes place. The rise in the concentration of the calcium starts the fusion of acetylcholine vesicles at the terminal membrane at the rate of 10,000 folds, and at each action potential there is 125 vesicle fuses with the terminal membrane of the nerve fiber and releases their acetylcholine into the synaptic cleft, where it is degraded by acetylcholine esterase into the acetate ion and choline, and then choline is reabsorbed for again start this new cycle, and formation of again acetylcholine takes place within axon terminal, and this all events occur within a period of 5 to 10 milliseconds. Fourth, the number of vesicles available in the nerve ending is sufficient to allow the transmission of a only few thousand nerve to muscle impulse. Therefore, continued function of the neuromuscular junctions, new vesicles need to be reformed rapidly. This is the molecular biology of acetylcholine formation and release. Now, the drugs that enhance or block transmission at the neuromuscular junction. Drugs that prevents the release of acetylcholine. Botulinum toxins is a product of bacterium Clostridium botulinum. Prevents the acetylcholine vesicles from fusing with the presynaptic membrane, thus preventing the release of neurotransmitter into the synaptic cleft. So this is the botulinum toxin. Botulinum toxin prevents the release of acetylcholine, and this is the product of Clostridium botulinum. Drugs that stimulate the muscle fiber by acetylcholine-like action and blocks the transmission. Many compounds, including methacholine, carbachol, and nicotine. Methacholine, carbachol, and nicotine have the same effect on the muscle fiber as does acetylcholine and act by causing localized area of depolarization of the muscle fiber membrane at the motor end plate where the acetylcholine receptors are located. They cause initial muscle twitching and then paralysis due to the persistent depolarization. Difference between these drugs and acetylcholine is that these drugs are not destroyed by cholinesterase or are destroyed so slowly that their action often persists for many minutes to several hours. These are the drugs methacholine, carbachol or nicotine. Their effects is same as the acetylcholine, but acetylcholine is degraded into the synaptic cleft and into acetate ion and choline is reused for the formation of the acetylcholine. But these drugs are not degraded as fast as the acetylcholine. So their effect persists for many minutes to several hours. Drugs that stimulates that neuromuscular junction by inactivating acetylcholinesterase. There are three particular well-known drugs: neostigmine, neostigmine, physostigmine, and disopropyl fluorophosphate. Third one is diisopropyl fluorophosphate. These three drugs inactivates acetylcholinesterase in the synapses so that it no longer hydrolyzes acetylcholine. Therefore, with each successive nerve impulse, additional acetylcholine accumulates and stimulates the muscle fiber repetitively. This activity causes muscle spasm. Muscle spasm, unfortunately, it can also cause death as a result of laryngeal spasm, which smother the person. Neostigmine and physostigmine combine with acetylcholinesterase to inactivate the acetylcholinesterase for up to the several hours, after which these drugs are displaced from the acetylcholinesterase so that the esterase once again becomes active. Conversely, diisopropyl fluorophosphate, which is a powerful nerve gas poison, inactivate acetylcholinesterase for weak, which makes this poison particularly lethal. Drugs that blocks transmission and the neuromuscular junction. A group of drugs known as cura-reform drug, cura-reform drug. This cura-reform drug can prevent passage of impulse from the nerve ending into the muscle. For example, D-turbocurarine blocks the action of acetylcholine on the muscle fiber acetylcholine receptor, thus preventing sufficient increase in the permeability of the muscle membrane channels to initiate an action potential. Myasthenia gravis. Myasthenia gravis is an autoimmune disease which occurs in about 1 in every 20,000 persons, causing muscles weakness because of inability of the neuromuscular junction to transmit enough signals from the nerve fiber to the muscle fiber. Pathological antibodies that attacks the acetylcholine receptors have been demonstrated in the blood of the most patient with myasthenia gravis. The pathological changes that occur in the neuromuscular junction in myasthenia gravis are summarized in this figure. These changes demonstrate why neuromuscular transmission is compromised in these patients. The end plate potentials that occur in the muscle fiber are mostly too weak to initiate opening of the voltage gated sodium channels, and thus muscle fiber depolarization does not occur. If the disease is intense enough, the patient may die of the respiratory failure as a result of several weakness of the respiratory muscles. This disease can usually be suppressed for several hours by administering neostigmine or some other acetylcholinesterase drugs that allow larger than normal amount of acetylcholine to accumulate in the synaptic cleft. Within minutes, some of these people can begin to function almost normally until a new dose of neostigmine is required a few hours later. These are the acetylcholine receptors in myasthenia gravis. This is pathological changes in myasthenia gravis, loss of functional folds, reduced acetylcholine receptors, and there is a accumulation of the acetylcholine into the synaptic clefts. This is the pathological changes occurs in myasthenia gravis has been represented before you through these diagrams. Muscle axial potential, a comparison with nerve action potential. Almost everything discussed in next section of initiation and contraction of the action potential in nerve fiber applies equally in the skeletal muscle fiber except for quantitative differences. A comparison of nerve and muscle action potential is resting membrane potential of the nerve action potential is minus 90 millivolt, whereas it is minus 80 to minus 90 millivolt for the muscle action potential. Duration of the action potential in milliseconds for the nerve it is 0.2 to 0.3, whereas it is 1 to 5 milliseconds for the muscle action potential. The velocity of the conduction in meter per second in the nerve action potential it is 70 to 120 meters per second, where it is 3 to 5 meters per second in the muscle action potential. The summary of events occurring at the neuromuscular junction. This summary express in neuromuscular junction. NMU is neuromuscular junction. Second one explain the structure, and third one is the drugs acting upon the neuromuscular junction. First, the action potential moves down the nerve. Second, opening of the voltage gated calcium channel and calcium enters. Third, neurotransmitter acetylcholine vesicle moves toward the active zone. Fourth, exocytosis and quantal release of acetylcholine. Fifth, acetylcholine binds to the receptor. Sixth, ion, sodium and calcium enter generation of the end plate potential. And seventh, end plate potential generates this action potential at the sarcolemma. This all things has been explained through the structure. This is the action potential comes at the terminal buttle. When this action potential moves towards the end of the nerve fiber, the opening of the calcium channels occur. Second reaction, opening of these calcium channels. Once the calcium channel opens, increase in the concentration of the calcium ion at the nerve ending, it starts movement of the acetylcholine vesicles to the periphery of the terminal. Then these acetylcholine vesicles fuse with the neurolemma and release their content acetylcholine into the synaptic cleft, which is the fourth action. Then acetylcholine binds at the acetylcholine receptor at the surface of the muscular plasma membrane, muscle plasma membrane. This occurs in the fifth event. And in the sixth event, the sodium and calcium generation enter of the sodium ion produces an action potential which is known as end plate potential at the muscle plasma membrane, and then 7 end potential generate the action potential in the sarcolemma. This is the seventh one. This all events explained through the diagram. Then it affects the effect of drug. Perisynaptic, first blockage of the sodium channels for the action potential by tetrodotoxin. Sodium channels can be blocked by the tetrodotoxin. Voltage gated calcium blockers is the conotoxin, and botulinum toxin also prevents the acetylcholine vesicles from fusing with the presynaptic membrane. Synaptically, acetylcholine esters, for the example, physostigmine and neostigmine, blocks the synaptically, and postsynaptically blockage on the acetylcholine receptor, for example, bungarodoxin, curare, depolarizing and non-depolarizing blockers. This diagram explains summary of events at the neuromuscular junctions in words through the diagram, and what are the blockers at the presynaptic level, synaptic and postsynaptic level. So the tetrodotoxin is the sodium channel blockers, conotoxin is the calcium blockers, and botulinum toxin is the acetylcholine vesicle blockers at the presynaptic level. At the synaptic level, acetylcholine esters, for example, physostigmine and neostigmines, are the synaptical blocker, and postsynaptical blockers are bungarotoxins. So this is all about the neuromuscular junctions, their structure, their function, and their blockers. Thank you for watching this video. Have a good day. Please do not forget, share, subscribe and like, which is our great expectation from you. We think that this topic becomes useful for my dear students.